muscle mass or tissue is made up of a large number of single muscle cells. The muscle cells are commonly called muscle fibers because these cells are long and slender in appearance. The muscle fibers are multinucleated and arranged parallel to one another with some connective tissue in between. The muscle mass is separated from the neighboring tissues by the thick fibrous tissue layer known as fascia. Beneath the fascia, the muscle is covered by a connective tissue sheet called epimysium. In the muscle fibers are arranged in various groups called bundles or fasciculi. The connective tissue sheet that covers each fasciculus is called perimysium. Each muscle fiber is covered by the connective tissue layer called the endomysium. Now, each muscle cell or muscle fiber is cylindrical in nature. The average length of the fiber is 3 centimeters, and it varies between 1 to 4 centimeters depending upon the length of the muscle. The diameter of the muscle fiber varies between 10 to 100 micron. The diameter varies in a single muscle. The muscle fibers are attached to a tough cord of connective tissue called the tendon. The tendon is in turn attached to the bone. The tendon of some muscles is thin, flat, and stretched, but tough. Such type of tendon is called the aponeurosis. Each muscle fiber is enclosed by a cell membrane called the plasma membrane that lies beneath the endomysium. It is also called the sarcolemma. The cytoplasm of the muscle is known as the sarcoplasm. Many structures are embedded within the sarcoplasm. Structures such as the nuclei, myofibrils, Golgi apparatus, mitochondria, the sarcoplasmic reticulum, the ribosomes, glycogen droplets, and occasionally lipid droplets. There are actually one or more nuclei in each muscle fiber. In long muscle fibers, many nuclei are seen. The nuclei are oval or elongated and situated just beneath the sarcolemma. Usually in other cells, the nucleus is in the interior of the cell. Now, all the organelles of the muscle fibers have the same functions as those of other cells. The myofibrils are the 5 parallel filaments present in sarcoplasm of the muscle cell. The myofibrils run through the entire length of the muscle fiber. In the cross-section of a muscle fiber, the myofibrils are seen separated from one another by the sarcoplasm. The diameter of the myofibril is between 0.2 to 2 micron, and the length of the myofibril varies between 1 to 4 centimeters depending upon the length of the muscle fiber. Now, light microscopic studies show that each myofibril consists of a number of 2 alternating bands, which are also called the sections, segments, discs. The 2 bands are, number 1, the light band or the I band. Number 2, the dark band or the A band. The light band is isotropic. When the polarized light is passed through the muscle fiber at this area, the light rays are refracted at the same angle. And so this band is called the I band or the isotropic band. The dark band is anisotropic. When the polarized light is passed through the muscle fiber at this area, the light rays are refracted at different directions. So this band is otherwise called the A band or the anisotropic band. In an intact muscle fiber, the I band and the A band of the adjacent myofibrils are placed side by side. It gives the appearance of a characteristic cross striations in the muscle fiber. A narrow lighter area called the H zone is seen at the middle of the A band. The I band is divided into 2 portions by means of a narrow and dark line called the Z line or the Z disc. The Z line is formed by a protein disc which does not permit the passage of light. The portion of myofibril in between 2 Z lines is called the sarcomere. The sarcomere is a structural and functional unit of the skeletal muscle. It is also called the basic contractile unit of the muscle. Each sarcomere extends between 2 Z lines of myofibril. Thus, each myofibril contains many sarcomeres arranged in series throughout its length. Each myofibril consists of an alternate dark A band and a light I band. In the middle of A band, there is a light area called the H zone. In the middle of H zone lies the middle part of myosin filament. This is called the M line. The M line is formed by myosin binding proteins. Similarly, the I band is divided into two equal portions by means of a narrow line called the Z line. The part of the myofibril between the Z line is the sarcomere. When the muscle is in a relaxed state, the average length of each sarcomere is 2 to 3 microns.